Now we looked at 3 different rats who had between 7 and 11 cells coming from the CA1 or subiculum region. So the environment was between 0.5 and 1 meter, and the recording duration was between 240 seconds and 1200 seconds. And first we compared 3 different spike sorting algorithms. So the manual sorting algorithm, like explained before, gave us an accuracy of about 14%. Then we looked at the cluster freak algorithm. This is an algorithm developed by David Redish, so it's a well-known algorithm that's very often used, which gave us 16% error rate, and our 3 sigma method gave us 15.6%. I forgot to mention this 3 sigma method. So basically, if we are creating templates of the different spikes for each neuron, and if a new spike is coming in, we compare to all the templates, and the template looking most similar defines the neuron ID afterwards. And additionally, we look at the standard deviation of the maximum and the minimum, and if the spike is outside the standard deviation, then we reject the spike because it's considered to be an artifact. So what we did here is we simulated the real-time behavior of the 3 sigma spike sorting algorithm. This means we used just the first 50% of our data set for calculating the templates, and all the other spikes of the testing data, the remaining 50%, were never seen before for anything. So this means it's a real-time test how well the spike sorting algorithm works, and you can see that the performance is almost the same as before. So we got 15.9% error. And then we can go on to the real-time experiment. So we acquire the spikes with the biosignal amplifier. We bend this filter the data to remove noise between 300 Hz and 6 kHz. We perform the real-time spike sorting, reject artifacts if they are outside of the standard deviation, and then we use the Bayesian algorithm to predict the position, and finally we compare it with the video tracking position. So here we have a video. This video is working, where we show the real-time spike sorting and firing field calculation. So like mentioned before, place cells were discovered 1971 by O'Keefe. Here we selected a very nice place cell of this specific rat, and we mapped it into the rectangular environment just for this movie. So if the rat is outside the firing field, you see that we are just recording noise and we don't see spikes. As soon as the rat goes into its firing field, the neurons start to fire, and you can see all the spikes here. We connected now everything to a real-time analysis system, and the system to a light, a LED. And this LED is only switched on if the spiking frequency of this specific neuron goes above 10 Hz. And you can see the algorithm is coding this firing field very sharply. So if the rat moves out of the firing field, it's switched off. Rat is moving around. And as soon as the head comes into the firing field again, the LED is switched on again. This means the spike firing is more than 10 Hz. Now we are playing it a little bit faster. And it's amazing how sharp the firing field is in this case. Now here are the results for the real-time tests that we did. So we had one training recording, 900 seconds long, where we did the spike sorting, we generated the templates, trained the Bayesian algorithm, and got an error rate of 14%. 1 hour later, we performed the first real-time experiment with data never seen before, and we achieved an error rate of 12%, which was even a little bit better than for the training before. On another day, we did another training, a little bit shorter, reached an error rate of 13%, tested it again in real time, and achieved 14%. And then we used the same training data set for another real-time run, where we had an accuracy of 17%. But what you can see is very important is that the offline result from the training is very similar to the real-time result. So this shows that the real-time position reconstruction can be done, that the whole system is fast enough to sort the spikes, to reconstruct the position. And normally the training data for our algorithms was acquired 1 hour before the real-time experiment. And in future it will be very interesting how long before we can acquire the training data, because learning effects and neuronal plasticity might change the firing fields. If we compare the results now to the other studies from China, who achieved accuracies between 4 and 10 centimeters with 30 to 25 cells in this figure 8 maze. Jensen and Lissmann, like mentioned before, achieved this 3 centimeters in this linear maze, 2 meters long, but they excluded intervals which had less than 9 spikes, and they also excluded foot stands with the large artifacts inside. If they used all the data for the reconstruction, they had also 15 centimeters error rate, which is exactly in the same range like we got in our real-time experiments with less cells. Braun, Barbieri achieved about 8 or 5 centimeters with 30 up to 40 cells. And Wilson, in the rectangular environment, had about 9 centimeters with a lot of 76 cells. So in our case, we had a rectangular environment between half a meter and 1 meter. And we divided this rectangular environment into 64 times 64 pins. So we had 4,096 possible positions for the prediction. And in the linear track, Lissmann and Jensen had only 150 pins. So there are 4 major differences for our study. We didn't exclude any segment of the data. We used everything. We had different training and testing data, which is very important. We had less cells. And of special importance is that the spikes were never seen before for the spike sorting, which is in contrast to the other publications. So they looked at all the spikes beforehand to do the spike sorting, and then they divided the training and testing data for testing the Bayesian algorithms. So all these conditions make the task more difficult, but it's very nice that it worked also in real time. So this was the first experiment that I wanted to show you. Are there any questions?
I just want to know what kind of errors you have. Are they false positives, or are they mostly errors where you identify a spike where there's no spike, or?
No, it's just a deviation from the predicted position from the real position, the error that we get.
And that's in centimeters.
That's either in percent,
Okay.
related to the diagonal of the
So
rule.
you don't know why, it just deviates. Just
It's a deviation from the real position. There are different reasons, artifacts, not enough spikes, wrong training data, whatever.
And the place cells fire in a predictable pattern within the space that's defined by the experimenter, or take up the entire space that's defined by the experimenter?
So normally you define the environment where you want to do the experiment, and then you place the red the first time inside. The red is moving around. The place cells are building up because the red is learning to know the environment. And after some hours or days of training, the place cells are stable, and then you can do this position reconstruction. And if you change something in the environment, you might destroy these old place cells. So it's depending how the environment looks like.